this overview where we're going to be first looking for an introduction. So what I aim to is give an overview about the problem that we will be looking at today, which is instant outbreak. Then I will cover some aspects of machine learning that are involved in the method and the objectives of the presentation itself. And after that, we'll be actually discussing the detecting outbreak based on pattern-based prediction method. So I'm looking in that direction because I'm in the second screen. So this is why I'm looking there, just to emphasize it. And during this part of the presentation, I'm going to be covering the methods, results, and the conclusions. So without further ado, I present this video from BBC, which is very helpful to illustrate what I mentioned about outbreaks. So outbreaks is meaningful to be explained here now, given the economical damage that caused. So imagine we have a moment when we have a population of insects with a higher number of individuals, very high abundancy. And when you had to these levels, as you can see in this video, some problems can start happening. So I mentioned about economical damage, but these scenarios can be occurring in different species as well, not only by locusts, that this example that I'm showing for you today. So that is one extreme event, as you can see by the amount of insects that we have in the screen in this video. But we can have different scales in terms of outbreaks, and that definition can be changed in different examples in agriculture. So the other examples that we can find as well as agriculture, there are other types of insects, more specifically different species, of course, that we're interested in studying. So we can look to different species and also have different definitions for them. So in agriculture, we can look, for example, this threshold that would be a number that will be decided by specialists that are studying that particular pest, and we might be mentioning what are these thresholds for this particular species. So for example, let's say we have one species and that will be its own threshold, that basically what this threshold will be informing to us will be once we reach that level and that population, we need to do some sort of intervention. So, for example, we can do biological control, we can do chemical control, or apply integrated pest management techniques in order to reduce the amount of insects that we have in that crop. So all of that is to reduce economical damage. So this presentation will be mostly focused on that example based on what we have in terms of data sets to illustrate for you. So that's about the context. And given that said, we have other techniques, other field of studies, it's in terms of machine learning and deep learning, that can be very helpful to actually work with this problem that I just presented. So before diving in the methodology, I just want to bring some topics on machine learning that will be useful for us when I will present the method itself and what it is about. So in terms of machine learning can be defined in multiple ways. Different books and research papers can define machine learning, but we can see as an area of artificial intelligence where we want to automate some specific tasks. So example of these tasks could be classification, could be regression, and in deep learning as well could be another area inside of machine learning where we're looking to deep neural network based algorithms. So we can be talking about that for an entire talk, but I will be covering in that level of machine learning. So when we say about learning, we have some definitions in mind and a couple of ways to define learning. So I will bring two of them here, just to illustrate what the method is based on. And these two types of learning could be related to one, the supervised learning, where we actually given some additional information to the algorithm. For example, here we have our grower very concerned about a lot of locusts that are in his crop, and he's very concerned about it. And let's say I want to collect some data when we have the outbreak itself. And all these data will have a label. So to me, it will be outbreak. And we have specialists saying, okay, this moment we have all these features, could be any feature related to, could be also the abundancy of the population that you're looking for. And we have the information that this data, these features are related to outbreak. And as well, when there's nothing happening, no outbreak at all, we can also inform the algorithm in this binary problem that I'm stating here. So we will bring both the data and the labels to the algorithm, and based on that, the algorithm will estimate its parameters depending on the machine learning that we technique that you're working with. And after that, we'll be doing the actual learning phase where we estimate the parameters based on these labels that will be already provided by specialists. On the other hand, we can have an unsupervised learning where we don't have the labels itself. We don't have that. We don't inform to the algorithm that this chunk of data is related to a outbreak, or that chunk of data is related to not outbreak, the way I'm stating here. So we don't get that, don't give that information. So for that, there's other feeds of algorithms that will be working to find different patterns and trying to cluster them, like hierarchical clustering, dimensional reduction techniques. Again, there's a whole few that we can be talking just for an hour or even more about it. There's active research on it. So it's a lot of topics to cover. But the main message that I want to give in this slide is to mention that we are working with supervised learning. So we say when we have the outbreaks and we say when we don't have the outbreaks in order to perform the actual task, which is the classification task. So the objectives, as I mentioned, we already kind of know, is to predict outbreaks. So we want to predict and build a new machine learning technique in order to predict an instant outbreak. So we want to do a classification task in that order. And in order to do so, I will present to you the method itself that we call pattern-based prediction, and we will illustrate the methods with a example with a population of aphids that our collaborators collected the data for us. And I will explain to you with benchmark that we've done with other machine learning techniques. So it will be three main messages here. First, we'll introduce the method itself. Then I'm going to show some benchmarking with other very common data methods that are used nowadays. Okay, so with that in mind, what's the data set itself? So the data set is about aphids. So aphids, there's another video that I'm presenting here from thanks to corporate. I can bring that video to you. So we have aphids. So aphids, it's very fascinating organism. This type of organisms have a very rapid development stage, given that. So we have lots of individuals in short period, a couple of days or towards months, depending on the species. And its development stage is basically you have two main phases. The first one will be when they stay in the plant, and then the plant will be consuming the phloem of the plant, as you can see in that part of the video. And there are all the other phase where they can fly and kind of spread over the crops. So it's very important in multiple, we can look in multiple perspectives and discuss the importance of these group of insects. But one of them is that they can transmit viruses and different types of diseases to the plants, and not only will cause economic damage, but will cause the economic damage based on that transmission of the viruses, and also will as well by consuming the phloem of the plant will actually be damaging the plant itself. So it's very important. It's a very fascinating system. Again, in terms of the example that we're bringing, there's a lot of biology that we can discuss about. But for now, I wanted to present a illustration of the method itself. So we have a data set on aphids. It's important parts of wet culture in Brazil. So thanks to our collaborators. And again, we thank a lot for that, because the data set here, it's very interesting. We have 108 observations, and you can see the aphids in the y-axis, aphids population, and weeks in terms of the x-axis. So as I mentioned earlier, I was mentioning about the thresholds that can vary for different species. So in this point here, when we're talking about the aphids that we're working with, our collaborators suggested 200 to be threshold for this group of aphids that we're working with. It's the total number of aphids that we're working with. So 200 will be the main threshold that we'll be using. So the only threshold. So that's presented here in the red line. Okay. So with this data set in mind, just want to emphasize some more details about it. So we have 4 traps in this monitoring system, and is weekly. So we have each observation is a week, basically. It's just the sum of all the aphids that were encountered in these traps. Perfect. So in order to present the methodology now, I will give some of the main concepts that we need. So we covered about the machine learning technique and supervised learning, and now we're going to be covering a little bit some of the metrics that we're going to be actively using. One, to do the performance of the algorithm, to check with the benchmarking, basically, and also with the one metric that is actively used to do the optimization of the hyperparameters that we use in our methodology. So first of all, this is a confusion matrix. The confusion matrix is based, we can read as when we have an observed data and we have predicted data. Predicted data could be from our algorithm in our case, but any algorithm. As we have a binary problem is a yes/no question. So we have a outbreak or we don't have an outbreak. So if the algorithm is predicting it correctly, we have true positive. So we have the frequencies of the times that the algorithm said that an outbreak would happen, and we observed an outbreak there. And the same thing happened when the algorithms predicted that there's no outbreak, and we observed that there are actually no outbreak. So we're happy with that. With the diagonal of this matrix, we are very happy. But when we look to the actually predicted saying yes, but observed says no, we have the false positive and false negative when the algorithm predicted no, and actually we have a outbreak, which is very, very bad. So we can actually have some metrics from it in our context. So based on true positive rate, false positive rate, and accuracy. So there are metrics that we can analyze the performance of the algorithm, and we can actually find these values and compare with different machine learning techniques. That's what it would be presenting in the end of the talk. So another interesting point, so these metrics will be used for actually for the benchmarking. And the main metric that we're actively using in the PBP method is the area under the rock curve. So area under the rock curve, we will have in the x-axis the false positive rate, and in the y-axis the true positive rate. So what I want, one way of reading it is if the method is really, really good, we expect that this area under the rock curve is 1. So it's very, very efficient algorithm and is doing the predictions very accurately. So that's one way that we can look at it. And our goal here is to use a area under the rock curve as an objective function. So I will be clarifying that when I cover the method itself in a minute, actually. So given that said, let's go back to our data, our time series. And in the time series, I just want to highlight some points that will be helpful for clarifying what the method is about. So we have in the hands that the red line is related to the threshold. It's 200. So once we reach 200, we need to do something about that. Basically, in the terms of practical terms, can apply any integrated pest management technique to it. So I want to predict these moments. And in order to do so, there's a paper in 2007 that helped enlighten an idea of looking to the patterns of a previous outbreak. So we have an outbreak here, and we try to figure out what are the patterns that happened before. So in that paper, they look not in a real data set as I'm presenting, but they look it into a Ricker model. So it's observations simulated from a Ricker model. And looking it back would be interesting to understand patterns. So they found the patterns that was very interesting, but now we are evolving in using the theoretical basis of that paper and applying to a machine learning technique to create this machine learning technique in order to do the predictions itself. So the point in observations before, we can translate it into a matrix what fashion, right? So we can say that X is the population abundancy, T1 will be the time that the first event happened. So what I mean by event is the outbreak. And -1 will be the previous observation. So have the moment of the event and the previous one, the second previous one, and to the nth observation. So n will be the observations that looked before. So we're just going back here. And what we're doing, so we have upper I outbreaks, and then we can store them all in this matrix and use the transpose for clarity. Once we have all this stored, we will have an overall idea of all the patterns. So possibly, and what we found actually, is that some of the patterns could be more related to each other, can have some clusters. So some of the patterns are more close to each other and others are close as well. So we try, just by looking to an entire matrix, we wouldn't be able to see it, all these clusters. So one of the parts of the process for the pattern-based prediction method is doing the clustering analysis. And for that, basically what we've done, we created this association metric. So the association metric is based on a come-beha distance, which is commonly used in count data problems. And the idea here is that we look to the patterns, right? Okay, so the pattern I compared to the pattern J, just saying that these are different patterns, Y is different than J. And what we're looking at is if the Canberra distance, that is a distance metric, is this Canberra difference between PI and PJ tends to infinity, our association metric will tend to 0. So it's very far. So we want that association to be close to 0. And the opposite, when the distance tends to 0, actually, we will have a association tending to 1, which I want they to have to be very close to each other. So we can group them using the proposed algorithm. And by grouping them and having all those clusters, we end up with the following picture. So as I mentioned, when I was showing the data, I said about a paper 2007. In that paper, they used the Ricker module for the simulations. So what I'm showing here to you today is the simulated values from a Ricker model, and this is the observations that we find based on that idea. So we have 5 observations before, so n is 5 in that case. And using n equal 5, we can actually use it to bring all these patterns. That will be the P matrix that I presented. And then we can cluster them into this 3 matrix. So I, in that case, would be equal 3. So I have 3 different clusters here that look based on our association. So now we have these 3 matrices, if you like. There are 3 matrices, and based on these 3 matrices, we can have an overall idea of what's happening per group. So in order to do so, we can bring another matrix that we'll be calling P prime means, which basically is the average of each one of the clusters in terms of each observation. And the observations 5 in the case. So the first line will be related to the first cluster, second line to the second cluster, and as a general example here, C in our example will be 3. So then we have a matrix that contain a average of each one of the clusters. So the average will be in terms of the observations here. And then we end up with our P prime means. Okay, so we have that P prime means will be a summary, but one important point is that each one of those clusters have different amount of patterns. So if we have one cluster that has loads of patterns in there, that's telling you something about it. It's telling you something that, well, when we have the outbreak, this type of patterns happen a lot of times. So that's an information that we can use to do the predictions. So each cluster, we have its informations, and we sum it into AP prime means. So the idea now is to provide another metric just to tell us now when are we going to say that we have an outbreak or not. So what we're calling here is d star pred as another value that will say to us it is or it's not a outbreak. So the idea is we have our P prime means matrix containing all the information of the cluster, so kind of a summary. And we have it individually stored the amount of patterns that we have per cluster. So as I'm presenting here, d star pred is a function of all these patterns. So the lines, L for lines or patterns, all the patterns that you find in each one of the clusters. And this function is written by the following. So we have a d base, d star base, plus this part of the function here. We are considering the lines, so the number of patterns. So I mean the lines of each one of those, number of lines here, number of lines here, number of line here, or the number of patterns. So just explaining the I thing. And here we have the L thing, and here we have the prime C and alpha. In order to explain it better, I prefer to show a picture. So let's say in the x-axis, we have 1 divided per the number of patterns in each cluster. And in y-axis, we have d star pred. And alpha here is showing different behaviors of that function. So it basically is saying when we have a lot of patterns, it tends to the d star base, which you can say that could be decided and will be optimized in the future. I will show you how. But we tend in there, and when we have not many patterns, you need to have a they need to, let's say a new observation comes in and it needs to provide you an answer. So this new observation needs to be very close to that cluster, like there's amount of patterns that are similar to each other when we have, for example, 2 or 3 patterns. So it needs to be very close to me to say, okay, we have an outbreak. And to decide, it must the association that we're computing must be higher than the d star pred. Okay, so a summary of all I've mentioned can be visualized based on this diagram. So we start with outbreak. So X is bigger than our X star here. So X star would be the threshold that depends on different species of insects and so on and so forth. We have this information. We have M, that would be the steps that we look previously of an event of the outbreak. D star cluster, that's the value that can change to group the clusters. And P will be our matrix, P prime C, so each matrix related to the clusters that we found based on the cluster. And then we have a summary that's just the average of each one of those clusters. Also, we use the D star pred, computing based on D star base and alpha, so that picture that I presented. And then in order to do an output, we first consider the P prime means and the value that we found for D star pred. And when we receive a new observation, we can say, oh, it's an alert or not an alert, based on the information that we gather from the time series. So we will apply the method, that's the method called pattern-based prediction. So we will be using it based on the patterns that we can see only with counting data. I'm just looking for the time series. I'm not looking to any other covariables. I'm just looking to the number of individuals now. So I mentioned to you that we need to optimize some values. So the learning in our case is supervised learning, and we need to optimize some sort of a hyperparameters. So this hyperparameters will be optimized in the following way. So let's say we have a combination of M alpha, and just showing M and alpha because I can visualize 3D plots. And the Z value here is the area under the rock curve. So that's what we're trying to optimize. So we're trying to find a combination of M, alpha, and also the star base in order that we have the maximum value of area of the rock curve under the rock curve. So we selected that. There's several ways to do it into optimization algorithms, and we selected to use the annealing-based algorithms called do annealing. And we also tested other types of another type of optimization algorithm based on genetic algorithms, and you can check in the paper. Actually, we did in the supplementary material, but we used the do annealing methods in order to do the optimization. So we optimize then based on the values that we obtain of the area under the rock curve. And to compute the area under the ROC curve, there is a selection that we do based on k-fold cross-validation. So let's say the training data set is 200 observations, and we can split the 200 observations into 5 groups, and then we train that group and test in order to find the area under the ROC curve, take the average of that. So that will be the value what we're trying to optimize in the end of the optimization or the training for the algorithm. So as I mentioned, for example, we have first 50% for training. That's when we do this optimization. I can go in more details if you have any questions. And we do for testing, we don't see this part of the time series, and we try to compute the metrics that we're interested in. So accuracy, true positive rate, and false positive rate. We use all of that and try to obtain our metrics to see if our method is performing well. And we did that with a different splits. So we started with 40, did the 50, and 60, 70, just showing the 50 and the 70 to give the idea. And we used as well 80%. So the pattern-based prediction is a method that will be using the patterns only solely of the time series in order to provide a prediction. And you imagine that we have other time series machine learning methods to predict events on time series. And here we tested, I will focus in a couple of them just to show first that we have the 40% train percentage, 50%, 60, 70, and 80. That's what I just showed in the image. The metrics, accuracy, true positive rate, and false positive rate. Okay, so that will be the metrics that we'll be using. So pattern-based prediction, and we have in parentheses false positive rate equal to 0, 1. So earlier I presented the one of the hyperparameter that will be our D star base, which these value could be selected in different manners. So one way we can select the value of D base where f, the false positive rate, is less or equal to 0.1. So it could be how much you accept to have a false positive rate. So how you're willing to have false predictions in that case. And as well, the pattern-based prediction with false positive rate equal to 0.2. And PBP, instead of using false positive rate and having values lower than 0.2, we can expect to have values of true positive rate that we want to have as high as possible, bigger than 0.8 and bigger than 0.9. So these are different variations of the algorithm that you can present. And one point that I wanted to highlight is here in this part of the comparison to have random forest with m equal 1, so just the previous observation, random forest with m equal 4, so we're just creating the data structure based on the previous observations, or 4 or 7, and trying to predict if we have an outbreak or not. And one thing that I wanted to say in an overview is that the method is very competitive with what we already have in literature. And in some cases, we will have for example, the true positive rate where we really want to have the highest as possible, higher than other points, for example, 0.75, 0.50, 0.75, 0.50. So it's a competitive method with random forest. We tested as well with support vector machines. So support vector machines is another technique, machine learning, very well used. And again, it's very competitive to the algorithm. And one of the points again that I mentioned, if you pay attention on the true positive rate of the support vector machine, it's not really high. And also the same apply to deep neural networks. So I mentioned earlier in the beginning of the presentation that we have deep learning techniques that are based on deep neural networks. So this is one implementation of deep neural networks based on TensorFlow, and we used to predict the outbreaks in the paper here. So overall, it's very competitive. And as well, the long short-term memory algorithm, that's a recurrent neural network algorithm. This is very used in the literature nowadays. And it's a variation. It's still a neural network based algorithm, which will have the idea of that methodology is try to, as well, using recurrency to predict the target of your problem. In our case, would be the outbreak. It of course depends on problem to problem, but then again, it's very competitive. Our method is competitive with what we could obtain with the other methods. And finally, one of the most important aspects of the methodology is its interpretability. So I mentioned all the patterns that we can obtain, and that's the main message. But what I want to emphasize is that when we use pattern-based prediction, we can go back in the patterns that were used for the prediction. And for example, we can use 25 to 75% precedential intervals in order to present based on those clusters of patterns that we found in the time series. We can bring in, highlight and bring in more information about what's happening given the previous observations that you're looking for the time series. So it would be useful as a visualization technique in this case here. If the black line is close to one of the groups that I'm finding here, we have 3 in this case, that to say if it's close or not, we can look back to the association that we're using, and that will tell us if we have an outbreak or not. So that's the main idea based on the previous patterns that we found. And this plot here is based on real data. So this plot here is based on the time series of aphids. So I want to emphasize. So finally, we showed the feasibility of our proposed method. Pattern-based prediction predicts population outbreaks based on time series data, high accuracy, and we illustrated a method predicting its past outbreaks using dynamics provided by aphids species. So we found its interpretability in the last, and it could, just in terms of time, I will stop about this main point here. But the interpretability in the paper, we have, I think, a paragraph just explaining how to interpret each one of the parameters and what's useful for. So in the paper we have more details, but the interpretability is a plus point of the methods compared with black boxes algorithms such as deep neural network. So I really would like to thank the Fundação de Amparo à Pesquisa do Estado de São Paulo, so Agência USP de Inovação, and Science Foundation Ireland Centre for Research Training for financial support, and my supervisors Rafael, Wesley, and collaborators Oliver, Edgar, Charles, Douglas, and Eduardo for all the help with this paper. And I have some of the references, and thank you so much. Thank you so much for your attention.